Attendees are in listen-only mode.
Well, hello everyone. My name is Neil Andrews and I am Executive Editor of the Pain Research Forum. I'd like to welcome you to today's webinar on the topic of pain epigenetics to be presented by Professor Steve McMahon and Franziska Hdeck. Today's webinar is the 7th in a new series of webinars that we actually launched last September, and we're really excited about today's event as the topic of pain epigenetics is capturing a lot of interest, and we have two great speakers and a great panel who've been doing lots of the work in this area. At the outset, I'd like to thank Hdeck and MedImmune for providing an unrestricted educational grant to PRF in support of the new series, and I'd also like to thank all of our general site sponsors for their support of PRF as well. In terms of the format today, Steve and Francisco will present a talk that will last about 40 minutes. After the talk, our panelists will take a couple of minutes to make a comment on the presentation, and then the rest of the time will be devoted to panel discussion and audience questions. Speaking of which, as always, we encourage everyone in the audience to submit questions to the panel at any time by typing them into the questions area of your control panel window. We want to get to as many questions as possible, and the panel wants to hear from you, so please send the questions in. One quick note, if you are ever having any trouble with the audio, if you're using your computer speakers, just make sure that the volume is turned up high enough. If you're still having any trouble, you should try the telephone. If you're having any trouble on the phone, you should try using the speakers on your computer. That usually solves any problem. So without further ado, now I have the pleasure of introducing our moderator, Professor Laura Stone. Laura is an assistant professor at McGill University in Montreal, Canada. Laura, welcome. It's great to have you here today. Please go ahead.
Thank you, Neil. I'd like to begin by thanking Neil for organizing this and the Pain Research Forum and our sponsors for making it possible. We're going to start by introducing our two panelists. Dr. Chaz Boutra is the Chief Scientist of the Structural Genomics Consortium at the University of Oxford. Prior to this position, he was Vice President and Head of Biology at GlaxoSmithKline. Dr. Santina Cianco is an assistant professor at the University of Catania in Italy. She is currently doing some very exciting work on the regulation of glutamate receptors in chronic pain. Today we have two speakers. The first is our team is Dr. Steve McMahon, who is the Sherrington Professor of Physiology at King's College London in the UK. Steve works to understand pain mechanisms using techniques ranging from molecular biology to electrophysiology to human psychophysical studies. With over 250 publications, he has made enormous contributions to pain research, including pioneering work on the potential therapeutic applications of trophic factors. He's currently focused on neuroimmune interactions and the genetics and epigenetics of pain. Dr. McMahon also directs the London Pain Consortium and is the academic leader of EuroPain, which is a collaboration of scientists working in academia and industry. Our second speaker today is Dr. Franziska Hdeck. Dr. Hdeck completed her PhD in neuroscience at Oxford in 2009 in neurodegenerative disease. Since then, she's been collaborating closely with Dr. McMahon at King's College London. Dr. Hdeck is particularly interested in understanding why certain people are more vulnerable to develop chronic pain than others. Her work is designed to test the potential role of epigenetic mechanisms in the hypersensitivity observed in chronic pain states. We're very delighted to have both Steve and Franziska today here to present their talk, Pain Epigenetics, Current Research and Future Challenges. Steve and Franziska, please proceed with your talk.
So, thank you, Laura. Fran and I are sitting here in London, and we're looking forward to interacting with whoever's out there listening to us. Before we do, we have one little bit of business on the slide I'm showing now, my disclosures. The lab here is funded from a variety of sources. We do quite a lot of work with private bodies, who I list here. However, I would say that all of our studies on epigenetics are funded by not-for-profit organizations. All that work is funded from charities and research councils. Right, so let me turn to the main topic of the day and tell you a little bit about what we're going to tell you. So we're talking about epigenetics and pain, and we realize that this is a relatively, it's a new field, certainly for pain scientists, it's a new study. In fact, before this webinar started, I looked up on PubMed I put in genetics and pain and got 14,958 hits, and then I put in epigenetics and pain and got 71 hits. And just to rub salt into the wound, I followed up and asked how many of those 71 were original, and actually nearly a half of them, 33, are review articles. So this really is, it's a field at its beginnings, and what we really want to do today is to, first of all, as you can see at this first point, we'll tell you a little bit about what epigenetics is, and I think that's probably important because it's likely to be unfamiliar to most pain scientists, and we'll talk about two principal forms of epigenetic modification. That's DNA methylation and a variety of forms of histone modifications. So first of all, we'll tell you what epigenetics is, and in that introduction, it will be longer than perhaps most introductions would normally be. We also want to tell you why we think epigenetics is an important field of study. And then for the bulk of the webinar, we'll turn to the issue of why pain and epigenetics. And here, as you can see, there are several different topics that we want to touch on. And just to introduce those briefly now, one important point we want to make is that these epigenetic processes do offer novel therapeutic targets which we hope might lead to an improvement in the treatment of pain in patients. And that could really start happening very quickly indeed. We'll also talk a little bit more generally and perhaps on the basis of even less evidence, concrete evidence, about other possibilities that make epigenetics a very important field. And those are the last 2 points on this slide. There's a kind of belief that some forms of pain become consolidated into a chronic state and may be more intractable to treatment. And epigenetics will provide a very ready mechanism for that horrible word, chronification. And a related but distinct feature is this last point, is that lots of pain scientists are starting to ask in the clinic, why do people with apparently similar diseases, injuries, experience very different amounts of pain? And is it true that some people are more vulnerable to develop chronic pain? And that too is something that's a very important concept. At the moment we have almost no framework of understanding that vulnerability, and as I hope we'll explain to you today, epigenetics could provide a very useful mechanism for understanding these properties that are not at all well understood but are clinically important. So that's what we're going to set out to do, and Fran's going to start by giving an overview of the basics of epigenetics.
All right, hello everyone. Thank you for joining in today. As Steve mentioned, I will give a brief introduction to what epigenetics is, and then we'll tell you more about how it's relevant to pain. First, very simple definition from the NIH Roadmap project. This was one of the first projects that really wanted to study epigenetics on a genome-wide level. And they define epigenetics as a process that involves the study of changes in the regulation of gene activity and expression that are not dependent on gene sequence. Classically, one would have looked at these processes in cells that divide. So one would have looked at these processes in terms of mitotic heritability. But more recently, this definition has been expanded and has also been expanded by NIH to include stable and long-term alterations in post-mitotic cells such as neurons. So to be a bit more concrete, we all know about what genetics is and how mutations in our genetic sequence can affect our lives and our children's lives, really. And now superimposed on that are these epigenetic mechanisms, such as DNA methylation, histone variants, post-translational modifications of histones. And many of these epigenetic mechanisms are fixed during development, but they are also thought to remain plastic throughout the lifetime of an organism, and they are thought to be one of the main mechanisms by which the environment can influence gene function. So for the purpose of this talk, we are going to focus mainly on DNA methylation and histone modifications. What is DNA methylation? DNA methylation is simply the addition of a methyl group to cytosines within the DNA. Most often this occurs in the context of a cytosine being across from a guanine across the phosphate backbone, and that's why it's referred to CpG methylation. Not all methylation occurs in the context of CPGs. There's non-CPG methylation, and not all CPGs are methylated. In fact, in promoter regions of genes, they very often have very many CPGs, but they're not very methylated, and these regions are referred to as CPG islands. Methyl groups are added to the DNA by enzymes called DNA methyltransferases, and they can be removed by TET enzymes in a process of iterative oxidation. Now, once our DNA has been methylated, it is then wrapped around quite tightly around histone proteins in the nucleus of our cells. And there's different types of these histone proteins, histone H3, histone 4, histone 2A and B, and they form these octameric structures that have lysine and arginine residues, which again can be modified by various chemical modifications like methylation, acetylation, phosphorylation, ubiquitination. And they can be modified in different positions as well. So for instance, here is lysine residue 4 of histone 3, and it has a single methyl group, so that would then be described as H3K4-methyl-1. And taken together, all these modifications, the sum of these modifications, is thought to affect the chromatin architecture, and so the wrapping of the DNA, whether the DNA is wrapped loosely or tightly, and they will also help recruit transcription factors or prevent transcription factors from binding. So obviously this is a very complex process, and I'm going to try and simplify the functional relevance of this process somewhat by giving you a few basics to remember. So there's really 3 basic facts you need to remember about epigenetic mechanisms. Firstly, epigenetics are crucial for normal development. Epigenetic mechanisms are what determine cell lineage. So they are what makes a neuron, or an osteoclast, a microglia. And so here's just an illustration from Wolf Reich, one of Wolf Reich's reviews, where you can see very early on in development is this phase of complete DNA demethylation and then remethylation, and in these phases you will have pluripotency genes that can be expressed for a short time, and then they'll be silenced forever and allow developmental genes to be expressed and differentiate cells into their different cell types. Second fact, epigenetic modifications are cell type specific. So this is an example from Siller et al. They looked at 30 different cell types, both embryonic and adult cell types, and also different tissue types. And they looked at their methylation states. And even on a global level, you can see this is a principal component analysis of the different CG methylation states. And each cell type clusters differently, and they sort of cluster together in terms of how closely related they are. So in vitro-derived embryonic cells cluster closely together, primary cells cluster closely together, and so on. But they're all different from each other. And finally, many of our epigenetic modifications are stable. So if you look here on the right-hand side first, these are histone modifications. This group studies the turnover of these modifications. And the blue line here indicates the cell turnover, and so you see many of these modifications like H3K9 methylation, H3K27 methylation turn over when the cell turns over. So they really don't change during the lifetime of a cell. And here on the left you can see an example of methylation. On the bottom here is the difference in methylation, so the figure of 1 would indicate there's a lot of difference in methylation, and 0 means no difference. And these people were looking at autosomal CPGs, and again they define, according to their definitions, about 22% of autosomal CPGs vary, and all the other ones are pretty stable across cell types and also across development. Also not M9, but 22% of autosomal CPGs in absolute terms is still 5.6 million. So that's still a lot of variation. And obviously we are very interested in the variation, because we think that the variation is what might be one way by which the environment can impact our genes, and is what we really want to study. So just a bit more on this variation. When does variation occur? So there's again 2 things that are quite important. Firstly, variation occurs preferentially at regulatory elements. So this is again from the Siller paper, and they look where do these CGIs occur that vary, and they occur mainly at promoter regions, at enhancer regions, and at these CPG island shores. These are regions adjacent to CPG islands that are often thought to contain alternative promoter elements and also are involved in cell regulation. And so secondly, not only does variation occur at regulatory elements, but it also seems that variation may increase over time. So as we age, we sort of encounter a lot of environmental experiences, and this may well then be reflected in our epigenomes. This is data from Praga and colleagues. They looked in twins, and they looked at global methylation and global acetylation levels. And you see in 3-year-old twins, there's very little difference because twins would have the same DNA and the same zygotic environment, so the epigenomes would be very similar. But then when you look at 50-year-old twins, there's really quite big differences that are starting to emerge. All right, so epigenetic modifications are setup specific, very important during development, stable, and when they do vary, they vary at regulatory elements and they vary over time. A last thing that I want to leave you with is that there is a wide range of modifiers that act on these epigenetic marks. So there's enzymes, big enzyme families that add these marks. They're referred to as writers. There's many proteins that bind to these marks and interpret them and recruit transcription factors. These are known as reader proteins. And then there's similarly big protein families that remove these marks, and these are known as erasers. So now I'm going to hand you back to Steve, who will tell you more about how we think this is relevant in pain.
Thanks, Francesca. So I realize that some of this might seem a little bit arcane, and what does it mean for a pain scientist? But I would like to start by trying to highlight why we need to study mechanisms like this. So, number one, this idea that they may be potential targets. So let me start with a fact. So this is some data that we published a couple of years ago. It's looking at transcriptional dysregulation in dorsal root ganglia in a rodent model of neuropathic pain. So in this case, the model is one of spinal nerve ligation. DRG are taken a couple of weeks later. Their RNA sequencing is performed on those DRGs, and they're compared to normal animals. And what you can see in this volcano plot is a large number of genes, either in red that are upregulated or green that are downregulated. And these genes, what they number, over 2,000 genes, show a fairly robust level of dysregulation and high levels of statistical significance in all these cases. So that's really what the pain field has been doing for the last 10, 15 years. It's been looking for candidate genes, and really, if you want to be a bit pedantic about it, all of these 2,000 genes could be pain genes. It's looking for candidate genes, and it's tried to say which ones of these are important for what aspects of abnormal pain signaling. And that's, it's a lot of work to do. I mean, to do a study in this one locus, in this one pain signaling site, the DRG, there are several thousand candidates. And of course, studying one gene at a time, looking at a knockout of this gene to ask whether it's the secret of neuropathic pain, in a way that presupposes that these genes act independently and that one or single genes will have large effect sizes. So I think an alternative would be to say, well, why don't we look at this as a more holistic process, and we look for the mechanisms that coordinate and drive these different patterns of gene expressions in chronic painful conditions. Those processes might give us better insights as to, I mean, there may be less of those mechanisms, so they're more targetable in terms of drugs, and that by regulating the process rather than individual genes, we may have a greater effect on the outcome of the whole system. And I think really that's my sales pitch as to why we should be looking at epigenetic processes as targets. They are the processes that lead to these different patterns of gene expression. And I think it's certainly, at least arguably true, I think the example I've chosen here is an extreme example, but I think it's arguably true, it's likely to be true, that in all chronic pain states there will be an associated genetic dysregulation taking place at a number of different levels of the neuroaxis. So this is really why I think generically these targets should be of interest to us. They are the things that change gene expression. Altered gene expression are the things that explain most of the pathophysiological properties we're interested in, changes in synaptic function and the like that occur in persistent pain states. Okay, so how tractable are they? That's the pitch, the plausibility. Well, they're actually very tractable because we already have tools to target particular epigenetic processes. And the one that has had the most attention in the literature, in fact I think the first paper came from Santina, one of our panelists, was to study this group of molecules, Hdeck inhibitors. So Fran has told you that we have things that write marks. So in terms of histone acetylation, and that's what these Hdeck inhibitors target, HATs are writers that add, they're histone acetyl transferases that add acetyl groups to histones, resulting in a more relaxed, open form of chromatin that is typically seen as being more transcriptionally active. Conversely, we have some erasers, Hdacks, which remove these acetyl marks, leading to a more condensed form of chromatin, which again traditionally is seen as more repressed. So Hdeck inhibitors are drugs that are able to block the action of the Hdacks. They will lead to a hyperacetylation of bits of chromatin and change gene expression as a consequence. Now the reason that these Hdacks have had a lot of interest is that they've been licensed for nearly 10 years now in the treatment of some cancers. So we have clinical experience in the application of these drugs to patients. And it's entirely feasible that we could apply that knowledge to the pain field. In fact, some of that is starting to happen. One of the problems, I would say, is that there are quite a number of Hdacks, and we don't have yet drugs that target individual subtypes of these Hdacks. I've tried to illustrate these blue lines here the specificity of some of the major Hdeck inhibitors that have been used in the clinic and indeed in experimental studies. And as you can see, some are really entirely non-selective in terms of their targeting. Others are more selective. These are the commonly used ones, targeting these so-called class 1 Hdacks rather preferentially. So these drugs are available. They've been used in a number of complex systems to try and examine the epigenetic contributions to underlying pathologies. So what about in pain? Well, we've done a number of studies, which I'll highlight a couple here, partly because I think they illustrate the principle that targeting some of these epigenetic processes may be of some clinical value. So here's some data from Frank's work, actually, looking at continuous intrathecal delivery of a couple of Hdacks into the intrathecal space of adult rats. And what we're showing here in these top bands of these westerns are the levels of acetylation, global acetylation of spinal cord taken at the end of exposures of 1 or 2 weeks of Hdeck inhibition. And we're looking at one locus, we're looking at H3, K9 acetylation, and the important point is that the drugs, MS30, MS60, lead to an increased level of acetylation globally in the spinal cord. And that's exactly what they should do if they're Hdeck inhibitors. So the drugs are doing what they're supposed to do. And the reason for telling you all this is that these drugs do have quite an impact on neuropathic pain development in models of nerve injury. So there's quite a lot of data on this one graph. In all of these experiments, the animals were fitted with intrathecal pumps here, day 0. The pumps were filled with either vehicle, that's the pink line, or with different amounts of different Hdeck inhibitors, that's the other lines, and they were delivered for a week continuously. And then the neuropathic injury was created, and the animals were studied behaviorally for several weeks following. And what you can see is that the vehicle-treated animals behave as expected from the literature. That is, after nerve injury, they show a very prompt, rapid onset, very persistent hypersensitivity, lasting at least several weeks, many months in most cases. You can see several things from the Hdeck treatments. The first point is that the Hdeck inhibitors on their own do nothing. So in this 1 week period, we're soaking the spinal cord in Hdeck inhibitors. It is leading to hyperacetylation of cells in the spinal cord, and yet we don't see any behavioral consequences in terms of a pain phenotype. So so Hdeck inhibition doesn't change sort of basal pain sensitivity. However, after the injury is created, all of these groups, and these groups encompass experiments that use different Hdeck inhibitors, subtly different models of neuropathic pain. In all cases, we see a highly consistent effect that is a very significant, modest 40–50% abrogation of the hypersensitivity that would normally develop. And we know that, all these experiments are done blind. Because we've done this experiment so many times, the result we are extremely confident of. The mechanism, that's another matter, but in terms of do Hdecks, do these represent a target? Well, clearly this preclinical experiment and others in the literature would suggest that they do. And I'd make one other point here, and that's that we think it's unlikely that these Hdecks are having very non-specific actions, just acting in sort of sedatives or something, because we don't see any of those effects in this control period, which would make us think that it's not a non-specific effect of the drugs. It's something to do with the drugs occurring in the context of injury that changes the outcome. So there's one approach, and I suspect that we'll see better compounds being made. I know there's been several reviews recently highlighting drug development in this area. There are quite a new series of new compounds that are selective for different classes of Hdeck, and several new compounds that are claimed to be relatively selective for particular isoforms. So that's one way that I can see this field developing. The other way, and I'll give you one second example here, I'll skip that one, is to use the power of mouse genetics to target individual readers or writers, epigenetic processes, try and assess their contribution to pain signaling. We've had a go with one example. We picked on Hdeck4 for several reasons, which are listed here. There's a little bit of literature suggesting that this is it's an Hdeck with neuronal functions that might have something to do with pain. And one of the reasons we were quite keen to pursue this matter was that in the experiment I told you about that looked at next generation sequencing in DRGs in neuropathic conditions, of those thousands of genes that were dysregulated, we ran some pathway analysis to see if there were patterns in the genes that were dysregulated. And we found that there were several nodes which were connected to many of the genes, highly statistically overrepresented in the genes that were dysregulated. And so two of those nodes are illustrated here, but Hdeck1 and Hdeck4 were connected in an unexpected way to many of the genes that were dysregulated in the neuropathic model. So this would at least give a clue that these might be regulators of the, they may be coordinators of the transcriptional change. So we've done quite a lot of work actually. Again, you're chipping away one target at a time. Maggie Crow did most of this work. She made 2 lines of transgenic mice. One line was a knockout of Hdeck4 driven by the promoter of NaV1.8, which is expressed in most but not quite all nociceptive neurons. And so this is a sort of nociceptive specific knockout of Hdeck4. And we know that we have knocked down Hdeck4 protein. The other line was another conditional knockout, but this was an inducible conditional knockout protein using the adrenaline promoter, which is expressed in all primary sensory neurons. So in both these lines, we're able to eliminate Hdeck4 from most, if not all, nociceptors. In the first case, this occurs late in development. In the second case, it's inducible. It occurs with tamoxifen treatment, which we give in the adult stage. What you can see on the rest of this slide is that the basal phenotype of both these lines is remarkably unimpressive. They have normal responses to acute noxious stimuli, either mechanical or thermal in these cases. And it says several things. It says that Hdeck4 is not necessary for at least the later stages of development of these nociceptive phenotypes because they're present in this NaV1.8 knockout. Neither is it necessary for the maintenance of the adult phenotype because of this phenotype in this adrenaline knockout. However, what we did find, we obviously ran quite a number of more persistent pain models in these mice. We didn't see a phenotype in the context of neuropathic pain, nerve injury associated pain, but we did see a convincing phenotype in inflammatory conditions, in this case using complete Freund's adjuvant injected into one hindpaw. So what you can see on the left is the effects of the knockout, the NAV 1.8 knockout, that's the nociceptor specific knockout. The grey line shows the normal hypersensitivity that emerges compared with baseline after CFA treatment, lasts for a couple of weeks. The pink line shows that that is considerably reduced, significantly reduced, when Hdeck4 is eliminated just from nociceptive neurons. And a rather similar profile is seen in the middle panel when the other line of Hdeck4 knockout was studied, that have lost Hdeck4 just from all sensory neurons. So that really, I think, is quite powerful evidence that Hdeck4 is somehow regulating the response of the neurons to inflammatory stimuli. And again, I mean, one of the points that I think we'll come back to is that it's very difficult to really predict cause and effect in this epigenetics, but what we can say is that associated with this loss of Hdeck4 function, not only was there a behavioral phenotype, but these cells show, that the DRG neurons show a less a less marked response to inflammatory stimuli. So this heat map on the right-hand side shows that the genes that are significantly dysregulated between the knockout mice and their wild-type floxed counterparts after an inflammatory stimulus, and this was on a pain card that surveyed about 50 common pain genes, and you can see there's about 7-8 genes that are statistically significantly dysregulated. And if I had to take a hint, lots of these genes seem to be downstream from things like NGF, so that you could imagine some of the inflammatory processes are attenuated in these cells in the absence of Hdeck4. And so that's actually quite a promising result that would suggest that targeting some of these readers or riders might have some therapeutic potential. Now time is getting on a bit, and I want to turn to the last issue we want to talk about, and that's give a couple of examples of how epigenetic processes may serve not just as drug targets, but also to increase our understanding about, I think, important aspects of chronic pain development. And the first example I want to give is this idea that that some long-term stable changes, in this case DNA methylation, might underpin a stable change in phenotype, in pain phenotype in people. And I'll tell you about one experiment that has been recently published that addresses that point. So the background to this experiment is a sort of ongoing collaboration of these groups at the bottom here, utilizing a resource that exists in London, and that's a twins unit at St Thomas's Hospital. So one of the things we've been doing for a couple of years is phenotype. We've been looking at the pain sensitivity, experimental pain sensitivity to heat pain stimuli of several thousands of these twins. Some of them are identical twins, some non-identical. And we've used that for several purposes, but one of the team, Jordana Bell, had a clever idea, and that was, amongst all these thousands of twins, to look for monozygotic twins, twins that have the same, they're identical twins, but the same, they start off with the same genome and probably the same epigenome, and look for twins that are discordant for their heat sensitivity. So she picked twins, pairs of twins, that fell off this line. And then she did a genome-wide analysis of the methylation status of all of the genes in the genome after doing immunoprecipitation of methylated DNA and sequencing. And then she identified regions that were differentially methylated in an association with the pain phenotype. And then to give it some credibility, she did a replication cohort, not in twins in this case, in just 50 unrelated individuals. And that study was a lot of work, it was very expensive, another issue about epigenetics, but it did produce a couple of very nice results, and the most significant of them was published in this paper about a year ago. And it reported that methylation around the TRPA1 promoter, TRPA1 being a well-known pain gene, was associated with this altered phenotype. So in this middle panel here, we're plotting, I think the black is one twin and the red dotted line is another twin, showing differential levels of methylation around the TRPA1 promoter. This is the key graph. Each line connects a pair of twins, and it shows the level of methylation of the TRPA1 promoter against their individual heat pain tolerances. And you can see that there's this negative correlation between tolerance and the levels of methylation. So I think that's quite an interesting case, that these twins would have started off in an identical fashion over their lifetime, and most of the twins who volunteer for these studies are quite old. They're certainly mature. The average age, I think, is 50 to 60. Over their lifetime, some of them have presumably changed their methylation pattern, and that's associated with the emergence of a very distinct pain phenotype. That is, that they are now differentially sensitive to heat pain stimuli. Of course, again, causation is difficult. For what it's worth, this panel on the right utilized another resource from this same study, and that's that several hundred of the twins, not just the 25 discordant ones, had skin biopsies, and those skin biopsies were subject to microarray analysis. So we were able to look at the levels of mRNA expression, presumably in keratinocytes, which do express it, versus the heat pain tolerance. And there is a weak but significant correlation there. So as I say, we don't actually have causation, but the reason I think this experiment is important is that it says that changes can occur in the methylation status of the genome. This is a perfect place to see it, starting with identical twins, and that is associated with a marked shift in phenotypic response to a very basic stimulus, a noxious heat stimulus. So I think you could see how this could act as a kind of as a model system to understand how people might change with time because of life's experiences in an epigenetic way to change their propensity to develop chronic pain. Now we'd like to finish, I think, with one other example, and I'm going to hand you back to Fran because this is an example of her ongoing work.
Right. So I am going to wind this up with a hot off the press example of what we're researching at the moment. So this is another way of getting at why pain might become chronic, and we have sort of pinched this from the immune field, where there's been a lot of interest in enhancers. Enhancers are regions of the genome to which transcription factors bind, and they are marked by these H3K4 methyl 1 histone modifications. And in immune cells and macrophages, it has been shown that while most enhancers are set from development, so macrophages will have their own individual enhancer profile from development, there's a small but significant number of enhancers that emerge de novo when you stimulate macrophages with different substances. So when you stimulate macrophages with TNF-alpha, you get a certain enhancer profile. When you stimulate IL-1 beta, you get an enhancer profile, and so forth. And these novel enhancers are called latent enhancers. And there's been a lot of interest around these latent enhancers because it turns out that they can stick around once the initial stimulation has been removed. So when you stimulate macrophages with interferon gamma, here in this region of the genome you have a latent enhancer that emerges, and this enhancer persists even when the interferon gamma has been washed out, and actually when you then re-stimulate with interferon gamma, the cell will respond more vigorously. So this is an example from the immune system where it seems that a cell carries a sort of memory where the cell was modified in a more long-lasting way and has suddenly gained a spot to which a transcription factor can bind in a more long-lasting fashion. And so now we wanted to see, can we apply this to some extent to pain? Could it be that in pain latent enhancers emerge and that they then persist and somehow help for pain to become chronic? And so we looked at this in microglia. Microglia are the immune cells of the CNS, and we know that they play a very important role in pain. So we did an experiment where we managed to isolate pure microglia from the spinal cord of mice after they had undergone nerve injury. And so this is some of the raw data. You see these black bits here. These are sequencing reads, and the more reads there are, the more binding of H3K4 methyl 1 there was. So these are basically enhancer regions. The top line is from a sham animal. The bottom line is from an injured animal. And you see in this region a novel enhancer appeared as a result of the injury. And this novel enhancer is upstream of a gene called CCL12, which is an important chemokine that's being linked to P2X4 receptors that are important for microglial function in pain. And when you look at the transcriptional profile of these cells, how do microglial cells change the transcriptional profile as a result of injury? You get genes that are dysregulated, and actually CCL12 is one of the genes that is quite heavily dysregulated, and 3 more of these top dysregulated genes after injury in microglia also showed novel enhancers. So we think this could be something very promising. This could be a mechanism that could explain how environmental influences like injuries and things like that could leave a lasting mark on the genome. Okay, so just to conclude, we hope to have shown you today that the study of epigenetics in pain can provide access to novel therapeutic targets. We think that it might provide mechanisms that may account for the chronic nature of pain, and it may also explain why certain individuals are more vulnerable than others. We are obviously still at the very beginning of this exciting field, and there are quite a few challenges. Just to already mention a few, we can then obviously discuss this more in the panel discussion. One big challenge is cell type specificity. It is very difficult to isolate cell types from complex nervous system tissues, and yet we'll all have to do this eventually. As Steve mentioned, we have problems with drug selectivity, but hopefully one of our panelists, Jas Bantra, and his Structural Genomics Consortium, they will also help with this, and many other chemists and drug companies and so on are helping to provide better compounds. And finally, there's this issue of causality, which marks are actually driving transcription and which marks are just. So thank you very much, everyone, for listening.
So thank you very much, Dr. McMahon and Hdeck, for that very interesting presentation. What we will do now is move to the panelists and ask each panelist to just comment for a few minutes on the presentation and on other issues that you might think are important to discuss or questions that you might have. So let's start with Chaz, please.
Well, I mean, I knew with Steve and Fran giving these presentations that we were in for a treat, and we had a treat. Steve, as everybody knows in the pain community, did some phenomenal work in the late 90s, mid-90s around neurotrophins, and as a consequence of that great work, of course, we've now got anti-NGS showing quite remarkable efficacy in patients. And it's great to see Stephen and Fran leading this area. As Stephen said, it's very early, but it holds a lot of promise. And I hope that these sorts of presentations will encourage more people to work in this area and catalyze our collective efforts so that we can try and realize the potential of these targets as new therapies for patients. So I had a number of questions, Laura, but I'll delay those until you suggest.
Okay, great. Thank you. I'd like to remind the audience that we are taking online questions, so please type your questions into the chat box that you see on your screen. So our second panelist is Santina. What would you like to comment for a few minutes?
Yes, I just first want to thank Stephen and Francisca for this excellent talk and for being so clear in such a complicated field. Yes, I think we have a lot of open questions for this field. I have a basic question, a comment I would like to ask to our speakers. We know that epigenetic therapy is going to be a very promising therapy. But do you think that it's good to think about this therapy alone or in combination with traditional drugs in the field of pain, or maybe also in other fields?
Do you like us to take that now, or would you like to come back to that?
Why don't you address the question now?
So I think it's actually a question that's affected drug development in the pain field, in all sorts of ways. So there are, for a long time we've been looking for a magic bullet which was able to target pain selectively with minimal side effects, and that's been difficult. I mean, it certainly nature has done it by making mutations in NaV1.7, but the medicinal chemists, much as they are very good, but we haven't really found very clean drugs that have had spectacular effects that have reached the clinic. So I think combinations will always be a possibility, and I noticed that in the cancer field, that's exactly what they're doing with Hdeck inhibitors. They're using them in combination with other drugs now. So I'm sure that will be tried out pragmatically by doctors who need more pain drugs. I mean, the need for more drugs is urgent, and so people will take whatever armory is available and use them as they can. So that's really dodging the question a bit, because it says, I think, what's going to happen in practical terms. I guess what you're getting at is that, is it conceptually, is that the way we should be doing it? Should we be just targeting single factors? And there, I think there's at least single factors have simplicity on their side. There are, as I think Fran explained, that there's quite a constellation of epigenetic enzymes that change the epigenome, and so we've got quite a selection actually. So it's not like, what my feelings are that some of the other drug targets in the arena at the moment, like kinases, they're very widely expressed in many different cell types. And it's difficult to see how you could get selectivity by targeting some of those ubiquitous enzymes. But these enzymes really are likely to be quite cell-specific, quite context-dependent. So I don't think it's beyond credibility to believe that individual markers might prove to be key determinants, key switches, key coordinators of transcriptional change that contributes to pain.
Could I also add to that, Laura? I think Santina's question is a great one, and with all chronic, complex, multifactorial, heterogeneous diseases, including chronic pain, it's likely that we're going to have to attenuate these 2,000 gene changes that Fran showed, or Steve showed. And just hitting one of those genes just isn't going to do it. I think many of us hope that by targeting some of these epigenetic proteins, we might hypothetically affect the expression of, let's say, 200 genes. And those 200 gene products go on to cause chronic disease. So potentially by having a selective inhibitor or modulator of one of these epigenetic proteins, you could actually affect 200 gene products. And so I'm hoping at least that by pushing some of these epigenetic targets, there will be less of a need for combinations in the clinic. But I'm sure there still will be a need.
So how far away are we in terms of drug discovery from getting an epigenetic drug for pain?
You know that that question is asked of all scientists every day. How far away? They always say, well, 5 or 10 years away. I think for a bit of reality there, I mean, it might be useful for you, Chaz, to say, I mean, you're someone who's trying to accelerate this process by going about drug discovery in a slightly different way. So how would you address that? How would you answer that question?
Well, I mean, I suppose Steve, I've often tried to sort of think of the past, and I'm just thinking, when did you first show that anti-NGFs were potentially going to be useful in chronic pain?
1994.
1994. Now, 21 years later, we have some remarkable clinical efficacy data, but we still haven't reached the market. And, and there are, I think some people are still concerned that the molecules that are currently being progressed by Pfizer and Janssen may well not make it, because people will be concerned about the side effects of them. So, 25 years later, we haven't, 21 years later, we haven't done it. If I think of the sodium channels, I mean, Stephen mentioned NAD 1.7, 1.8, etc. Again, Stephen, when was the first paper from John Wood on the sensory neuro-specific sodium channel? It was early 90s. And the latest we know is that a number of companies have been working on that, and Convergence have some efficacy data, but that's just phase 2 data. It's still got to go through phase 3 and launch, etc. So again, you're talking 2 decades away. I think with the epigenetics, Stephen highlighted nicely at the start that this is a very early field. We're lacking lots of tools, etc. So I suspect, pragmatically, even if we had a target today, it would take us at least 10, 12 years to get it to the market. Now, we don't have a firm target today, so I suspect in terms of having an epigenetic drug for pain, I think it might be realistic to say that that's not going to happen for 15 to 20 years.
What about repurposing of other drugs, Chaz? So, I mean, because there is, I mean, pain has been a bit slow to pick up on epigenetics, but other disease areas have been embracing it more wholeheartedly. And of course,
Absolutely.
any gains they make, we can transfer, we can repurpose those drugs very quickly if they have efficacy.
Absolutely, and of course the challenge we've got at the moment, and the examples you highlighted with the Hdeck inhibitors, a lot of the molecules that are out there at the moment are very non-selective. And somebody could say, for example, develop an Hdeck4 selective inhibitor, then it might be worth taking a shot with it. So you're absolutely right. If we can take something that's already out there for a different indication and try and repurpose it, then that certainly will bring down that 15 to 20 years, and hopefully we can make it happen in a decade or so. But I suspect it's not going to be 5 years, because even if we had a molecule today, to do the phase 1, 2, 3 studies and launching it, it would take at least 4 or 5 years. So, I think realistically it's going to be at least a decade away. Now, in terms of Stephen said, what are we trying to do? And I don't want to talk for too long, but from a target discovery perspective, I mean, we all recognize that in academia there's phenomenally smart people like Stephen, Fran, Santina, Laura, etc. But what's lacking in academia is high-quality novel inhibitors or novel tools. A lot of those tools, of course, exist in industry. So what we've chosen to do is, within the SGC, is we've decided to pool resources. So we're working with 10 large pharmaceutical companies. We have access to their medicinal chemistry and their compound collections and their sort of assays, etc. We only work on novel proteins. So Stephen and Fran have talked about the Hdecks. We're working on other families of proteins, the demethylases, the bromine domain proteins, etc. And then we're working with these 10 pharma companies to generate high-quality inhibitors. And these are really high-quality inhibitors. I mean, for example, in the bromine domain family, there are 61 proteins, 61 bromine domains in humans. We've got purified 48 of them. So when we generate a molecule, we screen it against 48 bromine domains. So these are high-quality, potent selective inhibitors. But then once we've generated these inhibitors, we give them away freely to people like Stephen and Fran, and what we're trying to do is we're trying to crowdsource science on these novel proteins. And, of course, and then people like Stephen and Fran will take them and test them in their assays, but equally people working in the inflammation space or the cancer space or the epilepsy space, they will take them and test them, etc. And so we're crowdsourcing science. The other thing we do is that all of the data we generate and all the reagents we generate, we immediately make them available to the world because we're trying to reduce duplication. One of the challenges we've got at the moment is that there's too much duplication in biomedical research. Because in the pain area, we're all aware of what's happened, for example, with substance BNK1 antagonists or tryptophan antagonists. You can imagine how much resources are exposed and you get to get a drug from it. So I hope that gives you a sense of what we're doing. So we're generating novel inhibitors for people like Stephen and Fran to test. We're focusing our efforts at the moment on epigenetic proteins. Not the Hdeck families in Oxford, we're focused on demethylases and bromine.
Thank you, Chaz. That is an amazing program that you are leading. It's really important, and thank you for doing that. I'd like to direct a question from the audience to Fran. It's about Hdeck expression, and the question is whether or not basal levels of Hdeck expression differ between individuals in the same way that DNA methylation levels differ. And if they do differ, Do you think that this could be conferring any differences in pain sensitivity or vulnerability, like the changes in methylation?
Well, I would say the short answer is we don't know. I don't think anyone has looked at that. And the problem with asking this question is actually that in people, if you wanted to know in people, you would have to presumably look in the relevant tissues. And that is usually the problem. So you would have to look in post-mortem brains and spinal cord tissues. So I don't know. I mean, it would be quite a difficult thing to test, I think. But yeah, if it was true, if there were different levels of expression, I would think that could have consequences for an individual. Yeah.
A related question is, do you think that there are different epigenetic changes associated with different kinds of pain?
We gave one example today that where we had a class 1 Hdeck inhibitors seemed to have some efficacy in treating neuropathic pain when applied to the spinal cord. We found that targeting Hdeck4 in nociceptors had efficacy not in that pain state but in inflammatory pain state. So yeah, I think there's likely to be an interesting mix. That's presumably true in other areas as well, I think, yeah.
Yeah, and given that there's going to be some implications for what epigenetic drugs might be useful for what pain conditions, which is a whole other level of complexity that we're going to have to think about moving forward. So we have another question from the audience about, it's a technical question, about what types of epigenetic studies could be done on brain and spleen tissue from non-surgical neuropathic pain models. And we'll throw that one to Steve.
Again, it depends. So are we talking whole brain, whole spleen? Then you're more limited. I am now a big believer. I mean, in a way, we all started. I think we all started from whole brain and whole tissue, and I guess we have to. And in some instances, that's still the only way. But I really think that it pays to invest the extra time and the extra effort and the extra money, try and really isolate the cell type that you're most interested in. Even actually for transcriptional studies, I believe more and more, because otherwise what we're faced with is trying to interpret, what do these changes mean? They don't just mean pain versus no pain. They also mean neurons versus neurons and macrophages and T cells, And so when you're looking at epigenetics, that's very difficult to disentangle. So, yeah.
Right, and we could take it even further to discuss epigenetic changes in tissues that are pain generators, like intervertebral discs or osteoarthritic knees.
Correct. Yeah.
So it's not even just the CNS; it's everything.
Exactly.
Maybe what the questioner was getting at, in a very polite way, is that most of the studies in humans, in fact, the Giordano Bell's TRPA1 methylation study, of course, that's done on blood. And that is the sort of standard, because that's a tissue that's accessible. But then Fran's just told you that a lot of the epigenome is very cell specific, cell type specific. So looking in blood might be okay, but I mean it really might not as well. It's dangerous. And of course there are ethical and practical reasons why that's the case in humans, but that's less so in our experimental animals. And I think the onus, as Fran has said, is on us really to up our game a little bit and to try and pick out whether or not blood is a good surrogate for DRG or brain. I suspect it's not.
Can I ask, related to that, Steve and Fran? I mean, Fran, I think you showed a slide where there were epigenetic differences, if you like, in in vitro cells or primary cells or long-term cell culture. What are the implications for what we do preclinically?
Yeah, I mean, I think that's a really good point. And we are looking at collaborating with people that, for instance, you can now make sensory neurons from induced embryonic stem cells. You convert fibroblasts into embryonic stem cells and then turn them into sensory neurons. Like you take them from patients and whatever. And the first question there is, well, how does this affect the epigenome? Because obviously you're de-differentiating and redifferentiating, and at the end you have sensory neurons transcriptionally. But do they have the same epigenome? And so there's, unfortunately, I think a lot of legwork to be done, where first of all we have to do kind of boring descriptive studies, comparing, you know, sensory neurons from mouse to sensory neurons obtained from embryonic stem cells in mouse and see how different are they and so on. So I think I'm really excited about this area conceptually, but I think unfortunately there's a lot of work to be done, and I agree with whoever was saying this at the beginning. I would love to have more people work in this area so that we can, you know, move faster.
Speaking of moving faster, here's a question from the audience about tools and what types of additional tools are really needed to push the epigenetics field forward.
I mean, again, there's actually several. I think that one of the biggest bottlenecks in studying histone modifications is the number of cells required to do genome-wide chromatin immunoprecipitation experiments. That's starting to change. There's a group in Israel now that have produced a paper that's, I think, quite believably shows that you can use lower numbers of cells to do immunoprecipitation. But up until now, I mean, you have to pool a lot of material to get enough. So I think improving chromatin immunoprecipitation, from my perspective, would be one big thing. And then the second thing that's also starting to emerge is using gene editing techniques that will allow you to remove methyl groups specifically from specific genes. So that's where you can now get potentially at causality. So you can actually use TALENs to direct histone demethylase or a DNA demethylase to your gene of interest, and you can remove the methyl group from that gene. And with that you can then test, is methylation at this gene actually functionally relevant for your model?
Oh, that is a great answer. Thank you. Does anyone else on the panel want to discuss additional tools that would be useful?
Well, it has been already said, more specific drug that could selectively inhibit specific Hdeck isoforms. Probably, I believe that Hdeck's are the most promising candidates for a drug, and probably we can get more information in cancer-related pain, and if we can get more selective isoform-specific inhibitors, that could be a great tool.
Do you think, Santina, it would be also useful to have better antibodies to allow us to do sort of things like chromatin immunoprecipitation, etc.? Because I sense for a lot of the marks we don't have good reagents.
Yeah, definitely.
Sure, I agree totally.
You know, one of the members of the audience has asked whether or not potential therapies that target epigenetic mechanisms, would they need to be delivered intrathecally, or do you think they could be given systemically? And I think the reason for this question is because so many of the preclinical studies so far have been focused on sensory neurons or spinal cord.
Yeah,
So
I mean, in a way, I don't think they would. I mean, almost certainly, a drug would the most efficient kind of drug would be a small molecule that would be already active, and so it would be given it systemically. Because the reason that we go to a lot of effort to target drugs to a particular place is to restrict their action. So to increase our understanding of mechanism a bit, we try and say, oh, let's just knock out Hdeck from sensory neurons, or let's just give our drug intrathecally. Because if we gave those drugs systemically, we really would have no idea where they were acting. So we do it as a kind of scientific tool, as a convenience, to increase our confidence in what we're doing. Whereas I think as a therapeutic tool, you wouldn't have to do that. Of course, it's a double-edged sword that the side effects we see are also limited because we limit the targeting of the process. So it may well be that I'm sure systemic small molecules given acting systemically would be the standard way forward in the clinic. I think it has to be that way.
Right.
Can I ask Stephen Fran? Sorry, Laura. I was just going to ask Stephen Fran. You showed a slide with increasing changes with age. Does that mean when we get epigenetic drugs that we want to test in patients, are we going to have to think about stratifying by age?
Well, I mean, I don't know. I mean, this is one study that sort of gets bandied around a lot, but I think there are sort of alternative explanations for this result as well, and I don't know whether it's really fully understood what exactly happens with age. Yeah, so I mean, I hope not. Let's put it this way.
Yeah, I mean, actually, don't forget, two unrelated twins at 3 months of age or 3 years of age, they'll have quite different epigenomes. I mean,
yes.
the neat thing about this experiment is it's a curiosity that comes with studying identical twins, that they have to start off being identical, and and that allows you to see how they change. But if it was all true, then the implication will be that we all start from a different place. That's our genetic and our initial epigenome, and we all, through life, we end up at a different place. And therapeutically, I mean, I do think it could be that some of us end up in a bad place in terms of the symptoms that we suffer. So there, you really might want to stratify people. It may be that pain vulnerability is an epigenetic phenomena, and that identifying that would be of fantastic benefit to people.
It's a really good point that that I'd like to lead into from that point, which is we've been talking a lot about epigenetic changes being something that's bad and that we need to reverse or fix. But epigenetic changes will be anything that has happened as a result of our experiences or our environment that has modified the genome. So there's a question here from the group, which is, what about the role of positive things such as lifestyle or social environment or diet on epigenetic mechanisms? And how do you see those fitting in?
That's a good point, and actually there are quite a few experiments also in other areas, I think neurodegeneration and stuff. They've done things like look at the effect of exercise on methyl marks and DNA methylation and so on. So yes, no, you can definitely have the reverse as well. You will have positive influences as a result of epigenetic type of thing.
Yeah, all of our drugs might be anhedonic as well. So
Yes.
they may fix your pain but leave you terribly depressed.
Well, we
yes.
might get, there might be drugs that...
Sorry, go ahead. I think it's quite a cool topic, talking about functional food that can really change, epigenetically change, make modifications, and it's going to be a very cool topic in the future. I agree.
There are people out there, a lot of people taking methyl donors, like Sam.
Exactly, and it's
So
it has been shown out.
Yeah, it would be very interesting to see if that is a good thing or not, because the preclinical studies suggest that Sam might be, like someone suggested, maybe good for pain but bad for mood, or maybe it'll be good for mood but bad for pain. So there's a lot of work to do with dietary supplements as well. So thank you for bringing that up. So earlier, Chaz, you mentioned that you had some additional questions for the panelists.
Well, I suppose what I was thinking, as Stephen found it closer to the literature, I mean, sort of what's the data with Hdeck inhibitors, say, for example, in epilepsy? And certainly when I was working in GSK, there was a sense, I mean, there's still a belief that most drugs that work in epilepsy work in neuropathic pain. And, and maybe some of the clinical studies in epilepsy might be a little bit easier. But I don't know what the data is with the Hdeck inhibitors in epilepsy.
I had a quick look just for this meeting actually, and I still think the only approved drugs are in lymphomas, and so there are a whole host, there must be a dozen phase 1 and 2 clinical trials in other immunological, neurological disorders. But none of those have certainly got to phase 3 or registration, or past phase 3 and are approaching registration. So I think, as I said earlier, I think the pain community have been a bit slow actually to pick up on this interesting bit of biology, and so we're behind. But there are some advantages in being behind actually, that you don't make the same mistakes as everyone else perhaps.
I mean, with Hdeck inhibitors, my feeling is that the preclinical data, they really seem to have work in most things. You know, they have reports that they work in Alzheimer's, that they work in Parkinson's, that they work in Huntington's. So I don't actually, no one really quite has a mechanism. So that's kind of why I also felt like maybe we need to take a step back and first of all understand the very basic mechanisms, and then we can try and understand what it is that these drugs do. Maybe they just kind of put in a kind of generic break into any, you know, big sudden change in the system. My guess is that right now, especially the compounds that we have right now, which target the very generic, ubiquitously expressed Hdeck 1 to 3, that they really affect a very common process, a process that is common to a lot of diseases.
I suppose the one thing that Fran, well, not worries me, I'm a little anxious about it, sort of, I mean, I do worry that one of the reasons as an industry we have failed, or we've not delivered as many new drugs as we would like, is because I think in the early 90s we got seduced by single molecular targets. You know, so in the early 90s we could do high throughput screens, we could clone proteins and develop sort of cell lines, etc. We had combinatorial chemistry and so on and so forth. And there was a sense in the industry that, you know, we were going to generate lots of selective tools for different proteins, and we would get lots of drugs. Now, clearly that has not happened. That has been a monumental failure. Now, and we all recognize that all the chronic diseases we're trying to treat, they are multifactorial, they're heterogeneous, they're very complex. There's a lot of heterogeneity in the patients themselves, etc. And we know that many diseases out there are treated with combinations of drugs or indeed dirty drugs. And so for me, the attraction with the epigenetics has always been, as we discussed earlier, is that hitting one protein may actually affect the expression of 200 genes, and those 200 gene products go on to cause a chronic disease. So I think, you know, earlier you said, so there's been the issue about going for a single target. Then there's the issue about which cell do we go for. Well, the fact is, in all chronic pain states, it's not one cell type we need to modulate. It's likely a number of neuronal cell types in the DRG, in the cord, in the brain, a number of immune cells in the periphery and centrally, including glia, etc., etc. And then, you know, you commented earlier about which specific mark on which gene. Now, you know, we can go down to that level of detail. So a single target, a single cell, a single mark on a single gene. My worry is that that might be the wrong direction. As scientists, we like to think like that because we like things black and white and so on and so forth. But somehow, how do we bring in all this sort of systems biology, network biology that's happening? Because that's what we're going to have to treat. Treating pain is not going to be treating a mark on a gene in a cell. It's going to be treating many marks on many genes in many cells in many tissues through many different targets.
No, I mean, that's a really interesting point, a really good point, and I mean, I completely agree with you. And actually, I mean, a lot of the data in epigenetics will end up being genome-wide data, and when you analyze these data in more detail, you really get the sense that it is networks of genes. It is systems biology that will be the key, will be one of the keys. But once you have sort of an upstream regulator of a network, it would then still be nice to be able to interfere with this upstream regulator specifically, as Santina was saying, with a specific drug target or with a molecular tool or whatever.
I also agree. It is important, specificity is important in basic research to understand mechanisms, but sometimes the experience tells us that so-called dirty drugs usually works better than very selective drugs. So I agree with both of you. From if we are going to look for a specific mechanism, we need very selective and specific drugs. But for pain therapy, maybe something that with a broad spectrum activity will be more indicated, as Chaz
Well,
said.
if we go back to Stephen's earlier point that we try and repurpose something that's maybe already out there. I mean, how do we accelerate that effort? How do we take an existing agent, which is being used in some patients, build a rationale or get a better understanding of at least what its sort of molecular selectivity potency is? And Steve showed a nice table with the different Hdeck inhibitors and which ones they hit, etc. At least come up with some sort of rationale like that. Come up with some rationale that it's likely to work in some pain setting, and there clearly is data that you have all generated, including yourself, Santina, that these compounds could be useful at least in some types of pain. And then how, if we can build that preclinical rationale, how do we do some sort of small, relatively cheap, quick clinical study? Because the thing is, one of the advantages of being in academia, I think we are more able to do something like that. Industry will not do it for lots of reasons. So, if we really want to be serious about repurposing, and if we really think that some of these Hdecks that are already out there may have some potential in some subtype of pain, how do we accelerate that effort? I don't know.
And if we are lucky, we can get information about Hdecks that are already used in clinical trials for cancer, and to get information in cancer-related pain, and to see if which of these Hdecks better works in this form of pain.
Well, that's a good point, Spencer. The problem is a lot of cancer studies, I'm not sure they even look at pain.
Exactly. It could be a good idea to, as a secondary endpoint to verify.
I agree that I think that a lot of medicine is serendipity, and so some patients being given a drug for one reason will notice something. That's the Viagra story. That's so you can get lucky and get onto something that's meaningful. But I'd also say that if you go back 20 years, gabapentin was being used as an anti-epileptic, and it wasn't long before neurologists started to use it off-label to assess whether or not it might be beneficial in neuropathic pain patients, because as you pointed out earlier, Chaz, that there was a kind of clear linkage that anti-epileptic drugs were likely mechanistically to have some benefit in treating pain. And I think, paradoxically, while we have so few pain drugs in the armory, then people are more desperate to try out things. So I do think that there will be efforts to try things out, and the gabapentin thing only took a couple of years, before it was then re-released, and accepted as an effective pain therapy, and still doing very well.
That is an excellent example. Another one is duloxetine, an antidepressant, which is now approved for neuropathic pain, and it's always a serendipity. Or I agree. It's sometimes it's a serendipity results that make us to understand specific mechanisms.
But Stephen, I mean, you've got so much credibility in the field. You've got so many clinical friends. How can we accelerate that gabapentin experience? We don't want to wait 10 years, etc. And I said, look, if we try and rationally design a novel epigenetic drug for pain, it could be 15 to 20 years away. You rightly said, if we can repurpose something, we can do it a lot faster, maybe 10 years. Well, how do we reduce that 10 years? I mean, I think it would be great if collectively we could come together to do something like that, because you're absolutely right, that's what's needed.
You can tell us, Chaz, didn't the NIH have a program that obviously there are thousands of compounds that have been shown to be safe in patients in phase 1 trials, but they've failed for whatever indication they were being given in phase 2 or 3. So we have lots of compounds that are at least relatively safe to give to patients, but haven't been tested in a very broad spectrum. I thought there was a facility to make those chemicals available for scientists to explore in this kind of repurposing environment. Is that not the case?
You're absolutely right. There is that ongoing with the NIH, and I know Pfizer's involved and Lilly's involved, and I think also in the UK with the MRC, I think AstraZeneca's involved, etc. But these initiatives have been around for 2 or 3 years. Why? Maybe it's happening, and I don't know, for which I apologise. But why don't some of us get together? If we really think there's this Hdeck inhibitor out there that we think has some likelihood that it may work in a subset of pain, then how do we facilitate that experiment? And there I'm talking not, a lot of maybe the molecules you just mentioned, Stephen, are molecules that haven't made it to the market. So they went into phase 1, 2, and they were dropped for whatever reasons. But I was thinking even in terms of molecules that are already in the clinic, they're on the market. Is there anything that we could do with those?
Money's a great incentive.
Well, so we're reaching the end of our time today, and that is really the biggest question, which is how can we facilitate and move these treatments or potential treatments forward more quickly. In the last couple minutes, I'd like to give each panelist and speaker the opportunity to just make 1 or 2 concluding remarks. And let's start with Chaz this time, please.
Thank goodness. Well, I think with Stephen onto it, I'm confident we'll get an epigenetic drug in the next decade. So I put my neck on the block. Don't let me down, Stephen.
And Santina, do you have any concluding remarks?
Yes, I also agree that epigenetic therapy will be a great opportunity for pain therapy, and still I believe that it could be an opportunity to change the expression of specific genes, and probably in combination with traditional drugs we can achieve a better result.
Thank you. Fran?
I just would like to thank everyone for signing up and joining in, and like to thank you, Laura, and Santina and Chas for also participating. It's really fun to discuss with several people.
Thank you. And let's give the last word to Dr. McMahon.
Well, I've also enjoyed it, and actually it is good to talk. As an academic, I actually have a different kind of prejudice. So I do, obviously, a lot of us do science because we want to see it applied in some way. Many of us do it because we're just curious. And I really believe that a deeper understanding of the biology will ultimately produce better outcomes for mankind, not necessarily for the reasons that we see. So I'm a great believer in, it's good to go digging and see what you find. And I think this is a great opportunity. That's what's happening in this field of epigenetics. It's novel, it's different. Who knows where it's going to take us? It might go nowhere, but I definitely think it's worth a little bit of exploration.
That is a beautiful ending thought. Thank you very much, everyone, for your participation. I'm now going to turn the panel back over to Neil for a concluding remark.
Thanks, Laura. Before we finish up, on behalf of PRF, I'd like to thank Steve and Francesca for a really wonderful presentation today. Thanks so much to both of you. We also thank our panel, Chaz and Santina, also for a great panel discussion, and our moderator, Laura, for doing a great job moderating the discussion. We also thank all of our attendees for listening to the webinar and for submitting your questions. We just have 2 quick final notes. A recording of the webinar will be available within the next day or so on PRF, and so if you'd like a replay, or if you have colleagues who might like to listen but weren't able to attend today, you'll be able to access the recording shortly on the PRF website. And second, please save the date for our next webinar, which will take place on June 16th from noon to 1:30 p.m. Eastern Time. And this webinar is going to be presented by Cheryl Stuckey on the topic of sickle cell pain. So with that said, we hope you enjoyed the webinar today, and we look forward to seeing you at future events. Thanks again, everyone, and have a great rest of the day. Bye-bye.
Bye.
Bye.